So good afternoon. My name is Tim Center. I'm a postdoctoral fellow in the chemistry department here at MIT. And today I'm going to talk to you about our technology we're developing in the lab called the Aryl Antibody-Drug Conjugates. I'm going to tell you why we believe this technology can make an impact in the bioconjugation field, as well as how we're using it to develop safer, more effective biotherapeutics to treat cancer. I'd first like to thank the Responde Center for their support over the past year, as well as introduce our research team led by PIs Steve Buchwald and Brad Pentelute, along with graduate student Chi Zhang and myself. All right, so if you're unfamiliar with what an antibody-drug conjugate, or an ADC, is, an ADC is a relatively new type of targeted cancer therapy. And these are medications that are able to identify and attack cancer cells while leaving healthy cells unharmed. And this approach isn't really a new idea. Paul Ehrlich was awarded the Nobel Prize back in 1908 for his work in immunity. And he first coined the term of a magic bullet treatment for cancer. And so although the science at the time didn't really allow him to fully realize this idea, what he meant by this was that if you were diagnosed with cancer, you could be given a medication that would be able to localize to the site of the tumor and contain a highly cytotoxic component that could kill the cancer cells without hurting the rest of your body. And so in this way, antibody-drug conjugates are able to act as these magic bullet treatments by recognizing tumor-specific antigens found on the surface of cancer cells. And through this antigen binding, the antibody, along with attached small molecule drugs, are able to be taken into the cancer cell, where they can be broken down, releasing the drug and leading to cell death. And this strategy has been successfully applied with two ADC therapies entering the market over the past 5 years. The first was Adcetris, approved in 2011 for Hodgkin's lymphoma, and the second was Kadcyla, approved in 2013 for HER2-positive breast cancer. And so in both cases, they're able to attach very highly cytotoxic small molecules to antibodies using what we call a Palladium linker group. And although there are only two ADCs currently on the market, this is an area that's really expected to grow rapidly over the next 10 years with over 50 clinical candidates and a projected market that's expected to exceed $10 billion. And so although these new ADC therapies have really begun to transform the standard of care for these indications in cancer treatment, linker technology, or how we go about attaching these small molecules to biomolecules such as antibodies, still remains a challenge for ADC programs. And so shown here again is Kadcyla, and highlighted in green is this Palladium linker group. And these linkers are not entirely stable in your body. And so over time, you have release of the drug systemically, and this can contribute to side effects, including neutropenia, neuropathy, and nausea. And these can be serious and sometimes even dose-limiting in nature. And so we asked ourselves if we could address some of these current challenges in ADC chemistry by either developing new types of linkers that would allow us to control their physiochemical properties, or perhaps develop a method to directly attach small molecules to antibodies, removing the need for a linker altogether. And so our solution is what we call Aryl Antibody-Drug Conjugates, or AL-ADCs. And compared to traditional ADCs, our linker technology features tunable physiochemical properties, and so this allows us to attempt to improve things such as stability and drug loading while remaining compatible with controlled release strategies and engineered antibodies. And so we hope that we'll be able to use this technology to develop therapeutics with a expanded therapeutic window compared to traditional chemotherapies and ADCs. And so our technology really relies on developments that we've worked on here both in organometallic chemistry and chemical biology. And we've used these to develop a simple protocol that allows us to selectively activate certain functional groups as small molecules as these air-stable palladium complexes. And as shown here is this orange powder. These complexes can be stored on the benchtop for months at a time. They're very stable. However, they're spring-loaded to react selectively with cysteine residues found on the surface of proteins. And so in this way, under physiological conditions, we're able to modify these very highly complex biological systems in just a matter of minutes. And so this is really a platform technology that allows us to give a wide variety of proteins new functions by directly attaching functional small molecules to them. And these can include things such as affinity tags, bioconjugation handles, fluorescent tags, as well as drug molecules and cytotoxins. And so looking back at ADCs, shown here are kind of the diverse range of small molecules that are available to us when we think about designing these therapeutics. And they vary in properties such as potency, polarity, as well as drug target. And so using our chemistry, we're able to directly attach these small molecules to antibodies in ways that were before either extremely challenging or even impossible to do. And so just as an example, this is a camptothecin derivative, so this is a natural product that was investigated as a chemotherapeutic. And we're able to activate this as our Palladium complex, shown in the picture below. And when we treat this with the antibody trastuzumab, in only 30 minutes under physiological conditions, we're able to rapidly synthesize our desired AL-ADCs with excellent control over drug loading. And when we look at the properties of these AL-ADCs, we see that we can easily tune things such as hydrophobicity by altering the nature of the Aryl group that we employ. And we also see improved stability both under acidic and basic conditions, as well as to external thiols and blood plasma. So shown on the right is a comparison of malamide linker with our AL-ADC. And you can see whereas with the malamide linker, you have release of nearly 40% of the drug after 7 days. With our AL-ADC, it's still 95% attached to the antibody. So in conclusion, we believe our AL-ADC technology is an exciting new approach that we hope can make an impact in how we go about designing biotherapeutics to treat cancer. At this point, we're currently, with the support of the Responde Center, working on our in vitro proof of concept studies in this area. And over the next year, we hope to move into in vivo disease models, as well as identify potential partners and collaborators to continue this work. So for more information, we have recent publications in this area, and you can also visit our website at pentelute.mit.com. And so once again, thank you to the Responde Center, and thank you very much for your attention.